guys, welcome to BIOGURU, to your own channel. Today we will discuss about the viruses. So first of all, what is a living thing? So the state of being alive, and what is a non-living thing? The thing which is not endowed with the life. So there are many things. Living things are protist, plants, animals, fungus, bacteria, and non-living things are water, soil, light, minerals, air, etc. So now life depends on physical and chemical processes, some physical processes and some chemical processes. The smallest organism called microorganism are arisen from less number of cells. So viruses are a unique group of biological entities known to infect every type of cell. And viruses are link between living and non-living. So viral infection includes algae, fungi, protozoa, plants, animals, and bacteria. So, viruses are not composed of cells and cannot be seen under a light microscope. Virus particles contains a single type of nucleic acid, either DNA or RNA. Then, in virus, the nucleic acids are enclosed in protein coat made up of lipids, proteins, and carbohydrates. Viruses do not exhibit most of life processes of a cell as they are inert but maintain genetic continuity through multiplication and undergo mutations. They are certainly more than lifeless molecules. That's why they are called link between living and non-living. They are just only particles until they are not attached with the living cells. So, viruses are best described as infectious particles. And the study of viruses is known as the virology. They are host specific, called as obligate intracellular parasite. Sometimes they are bacteria specific, sometimes they are plant specific, sometimes they are animal specific. So, some MCQs. The study of viruses is known as bacteriology, biotechnology, microbiology, or virology. Yes, it is virology. So, which are the obligate parasites? Bacteria, viruses, fungi, protozoa, or it is viruses. Then, what are best described as infectious particles? Fungi, bacteria, virus, or protozoa? Yes, it is virus. That's nice. So, now discovery, classification, and structure of viruses. So, discovery. In 1892, the Russian pathologist Dmitri Ivanovsky, while studying tobacco mosaic disease, he filtered the sap of diseased tobacco leaf, and this infectious agent passed through the pores of the bacterial filter. Even they pass from the bacterial proof filter, and the filtered sap was injected to healthy plant. The symptoms of mosaic disease were seen in it. And Martin Beijerinck concluded that the disease causing agent was a contagious living fluid. So, Contagium vivum fluidum. W.M. Stanley crystallized TMV, tobacco mosaic viruses. And Frank Conrad confirmed that the genetic material of the TMV is RNA. So now classification of viruses. The ICTV regulates the norms of classification and nomenclature of viruses. What is ICTNV? International Committee on Taxonomy of Viruses. So the ICTV scheme has only three hierarchical levels: the family including some subfamilies, genus and species. And the viruses are named after the disease they cause, just like polio virus, then HIV virus, and then coronavirus, COVID virus. So the family names end with the viridae. The genus names end with the virus. So the species names describe their nature. And the ICTV hierarchy is family, then genus, and then species. So, classification of AIDS virus. Family is Retroviridae. Then genus is lentivirus. then species is human immunodeficiency virus, HIV. Now structure of viruses. Viruses range in size from 300 nanometer in TMV to 20 nanometer as in parvovirus. Then helical viruses, these are some structures. Helical viruses resembles long rods that may be rigid or flexible. Rigid viruses, rabies virus. Flexible viruses, TMV, tobacco mosaic virus. So these are the structures, rabies virus and TMV. Now polyhydral virus. Many animals, plant and bacterial viruses are polyhedral or many-sided viruses. For example, herpes simplex and polioviruses. So enveloped viruses. These viruses are roughly spherical. For example, influenza virus. So then bacteriophages. The viruses which infect the bacteria are called bacteriophages. Bacteriophages are not bacteria. These are viruses that can kill the bacteria. They have complicated structures, are called complex viruses. They have polyhedral symmetry in the head, helical symmetry in the tail sheath, and the envelope contains glycoprotein. Projections known as the spikes. So, spikes are useful for attaching the virus to susceptible host cell. So, it can attach with the susceptible cell. For example, measles virus. So, it is the structure of measles virus and it is bacteriophage. Now, all viruses consist of core and capsid. In core, they have genetic material, DNA or RNA, and capsid, that is outer covering. So, core is nucleic acid that forms the genome, and capsid is surrounding coat of the protein. The capsid gives the shape and protects the genome, and the capsid made up of protein subunits called capsomeres. So these proteins or subunits are called capsomeres, and the core contains genetic material, DNA or RNA. The viral genome contains double-stranded DNA, dsDNA, or single-stranded DNA or ssDNA. So viruses that infect plants have generally single-stranded DNA. So viruses that infect animals have double-stranded DNA, and bacteriophages have double-stranded DNA. Bacteriophages can kill the bacteria. So viral nucleic acids molecules are either circular or linear. Most of the viruses have single nucleic acid molecule, and few viruses have more than one nucleic acid molecule. HIV, human immunodeficiency virus, has two identical molecules of RNA representing its genomic copies. So it is the structure of HIV virus and papilloma virus. So TMV, tobacco mosaic virus. So tobacco mosaic virus is about 300 nanometer long and 18 nanometer in diameter. Its molecular weight is about 39 to 10 power 6 Dalton. Then capsid is made up of 2130 capsomeres. So 2130 capsomeres makes the capsid. So the capsomeres are identical size arranged in helical manner around a central hollow space of 4 nanometer, that is 40 angstrom. Each capsomere is made up of single polypeptide chain with 158 amino acids. And inside the protein capsid, there is a single stranded spirally coiled RNA molecule consisting of 6,500 nucleotides. Now, bacteriophages. Bacteriophages, for example, T4 bacteriophage. T4 bacteriophage can distinguish into head and tail regions joined by a collar. So, tail includes pins and tail sheath, base plate, and tail fibers. So, it is the structure of T4 bacteriophage. And the tail helps virus attach to host cells. The tail sheath aids injecting viral DNA into the host cells. So, it attaches with the cell and inserts its DNA or RNA into the bacteria. So, now some MCQs. Who crystallized TMV tobacco mosaic virus? Then Martinus Beijerinck, then Frankel Conrad, W.M. Stanley, or Ivanovsky. Yes, it is W.M. Stanley. So the viruses resemble long rods are helical viruses, polyhydral viruses, enveloped or bacteriophages. Yes, you are right, it is helical viruses. What are the examples of enveloped viruses? Herpes simplex virus, polio virus, TMV, or influenza virus. Yes, you are right, it is influenza virus. The virus which infect bacteria are called bacteriophages, helical viruses, polyhedral viruses, or enveloped viruses. Yes, it is bacteriophages. Spikes of bacteriophage are useful for the attachment to host cells, digestion of host cells, penetration or protection. Yes, it is for the attachment.